This is a race where we all could be the winners, the prize being the taming of the pandemic. Last week, the Morrison government made a preliminary bet. It's hoping Australia can manufacture the vaccine being developed by the University of Oxford and the drug company AstraZeneca.
This agreement that the government has made with AstraZeneca is a letter of intent showing that they are interested in this vaccine and that they don't want to miss out on the opportunity should this vaccine prove to be safe, immunogenic, and actually protect against COVID-19.
The Oxford COVID-19 vaccine has shown that it produces what looks like a good and a safe immune response in early animal and human trials. Now it's in phase 3 studies around the globe to prove effectiveness and safety in the real world.
In these studies, thousands of people will be randomized to either get the vaccine or to not get the vaccine, and they will see if the people that got the vaccine are less likely to contract COVID-19 disease.
CSL is a global vaccine manufacturer based in Melbourne, and while they're already working on another vaccine with the University of Queensland, they also have the capability to make the Oxford vaccine as well.
Look, I think they know how it's going to be manufactured. What they haven't done yet is really scale that to a level where you're able to produce, 50, 100 million doses so that you can vaccinate populations, widely.
Every COVID vaccine aims to train the immune system to attack the spikes around the virus. Leading candidates such as the Oxford and UQ vaccines do this differently, but both use recombinant or synthesized DNA. The Oxford vaccine carries a genetic code on a chimpanzee virus. When injected, that chimpanzee virus invades the cells of our body, and the code it carries instructs our cells to make the COVID spike proteins, which then trigger an immune response. The UQ vaccine cuts out the middleman. They've genetically engineered part of the spike protein itself and hitched it to an immune stimulant called an adjuvant. The vaccine then triggers an immune response directly.
So really very simple, one yields a protein, one yields a virus. And we have to be very careful that those two vaccines aren't cross-contaminated. So we will have a very strict process in place that ensures that doesn't happen.
A major impediment to the Oxford vaccine might be that it may have to be stored and transported at a much lower temperature than other vaccines.
Because these are virus particles, they often have to be transported at minus 70 degrees and so kept very cold. So I would hope that they might be able to develop something that can be stored in the fridge in the longer term, because obviously having to keep things at minus 70 degrees does have its constraints in many parts of the world that wouldn't necessarily have those facilities.
For the UQ vaccine, we're certain that the cold chain is going to be a 2 to 8 degrees storage temperature, and that will retain the stability of that vaccine.
The UK's Chief Medical Officer has been reported as saying that a vaccine might not be available until a year from now. It's a sobering thought, but Professor Katie Flanagan is optimistic.
We would anticipate that there should be some kind of signal and interim results coming through probably in about 2 months' time. And certainly by the end of the year, I would expect that we would have a very good idea.
Professor Flanagan has worked with the Oxford vaccine technology in the past and has been impressed with its safety.
I've actually been working on chimpapanoviruses myself, and we know that in all of those there's no concerning safety signals. So I think this is other encouraging data to suggest that this vaccine will turn out to be safe in these large-scale studies.
Hi, I'm Lee Sales. Thanks for watching this story. If you'd like to watch more of 730's stories, they are on the left of your screen. And tap on the button below to subscribe and get the latest from ABC News.